Hello class, today we're going to talk about nucleus and DNA. What? This is so interesting.
We're going to start with the nucleus. The nucleus is a large membrane and closed structure that contains genetic material in the form of DNA. Protons and neutrons form the nucleus, and it is the center of the atom. You should think of the nucleus as the boss of a big company because it is the control center of the cell. It contains nearly all the cell's DNA and coded instructions for making proteins and other important molecules. The nucleus is surrounded by a nuclear envelope which is composed of two membranes. It is dotted with thousands of nuclear pores. These pores allow materials to move in and out of the nucleus. Examples of such materials are messages, instructions, blueprints, a steady stream of proteins, RNA, and other molecules. So in this picture, we see the nucleus. And this right here is the nuclear envelope, and these dots are the nuclear pores through which information goes in and out. In the nucleus, you can find chromosomes. They carry the cell's genetic information. Chromosomes are thread-like structures which are composed of a protein and DNA. They are spread throughout the nucleus in the form of chromatin, and chromatin is a complex of nucleic acids and proteins. During cell division, its chromosomes condense, and we can see them under a microscope. Cells fall into two categories depending on whether they have a nucleus. These two categories are called prokaryotes and eukaryotes. In eukaryotic cells, the nucleus separates genetic material from the rest of the cell. The prokaryotes don't have a nucleus, but they do have DNA. Now I'm going to talk about the role of DNA. The DNA must be capable of three things. Firstly, the genetic material must store information needed by every living cell. For example, the genes that make a flower purple must somehow carry the information needed to produce purple pigment. Secondly, before a cell divides, its genetic information must be copied. And thirdly, when a cell is divided, each daughter cell must receive a complete copy of the genetic information, and this is called transmitting information.
Hello, today I'm going to talk about the structure of the DNA molecule. The DNA is made of nucleotides connected with covalent bonds into a long chain. These nucleotides contain three basic parts. 5-carbon sugar coat deoxyribose, which gives the name of the DNA molecule, phosphate group, and a nitrogen base. There are 4 types of nitrogen base, called adenine, thymine, cytosine, and guanine, which are marked with the first letter of their names. They are connected by bonds outside of the nucleotide chain. They could be joined in any way, and each sequence gives different and unique coded genetic information about the DNA. For example, ATC-GTT carries the information that the body has blue eyes. Also, the nitrogenous bases are good at absorbing UV light. And now I'm going to tell you more about the people who examined the structure of the molecule of the DNA. The Austrian-American biologist Erwin Chargaff found out that there is almost the same amount of adenine and thymine in the molecule of the DNA, and the same is with guanine and cytosine. Another person who worked on the structure of the DNA is the British biologist Rosalind Franklin, who started her project in 1950. She used a special technique called X-ray diffraction to examine the structure of the molecule. In other words, she applied an X-ray crystallography to find out more about the properties of the molecule. In 1952, her work resulted in an X-ray photography. While she was working on her project, other 2 biologists named James Watson and Francis Crick were working on a three-dimensional model made of cardboard and wire of the structure of the DNA, but their model wasn't able to explain in a good way the properties of the molecule. In 1953, when Watson saw the X-ray X-ray photograph made of Franklin, he was able to discover the double helix model. The double helix model is a model of the DNA molecule in which two strands are twisted around each other. The model basically looks like twisted leather. The two strands go in opposite directions and they're anti-parallel, which allows them to have contact in the center of the molecule. Another things that Watson and Crick found out about the structure of the molecule is that they discovered that hydrogen bonds are used to connect the nitrogenous bases. Even though they're weak, this is good because if they weren't, the molecule wouldn't be able to split. And another discovery from Watson and Crick is that they discovered the base pairing, which states that a perfect fit can form only between adenine and thymine and between cytosine and guanine.
Hi class, I'm going to talk about the DNA replication. So, before a cell is divided, the DNA should be copied. And this process of copying is called replication. So, the process starts with separating the two strands of the double helix into two separate forks. And this is with the help of the exome, which is called helicase. So, as each new strand forms, new bases are added following the rules of the base pairing. And for example, if base on the old strand is T or thymine, we should add to the new strand adenine, which is A. And this is the same with the C and G, cytosine and guanine. Woo! And if we have a base sequence TACGTT, it goes like ATGCAA, the opposite. So, a conclusion. The two DNA moles are initial to each other and also to the original one. And after the replication, there is one original strand and one new one. The principal enzyme involved in DNA replication is called DNA polymerase. So this is a keyword, and you should write it somewhere. It joins individual nucleotides to produce a new strand of DNA. And it also makes sure that they're almost perfect to the original one. Telomeres. Telomeres is called the DNA at the tips of the chromosomes. This type of DNA is difficult to replicate. In these situations is used special enzyme called telomerase. It is dividing the cells and preventing the genes from damaging. DNA is also found in the prokaryotic cells in its cytoplasm and in the nucleus of the eukaryotic cells, where it's packed into chromosomes. Replication in most prokaryotic cells begins at a single starting point and proceeds in two directions until the entire chromosome is copied, while in eukaryotic cells, replication proceeds from multiple starting points on the individual chromosomes and ends when all chromosomes are copied.
It was so interesting!